Wonderful. I'm, speechless, which is a difficult thing to achieve, apparently. I'm going to talk about, receptors, Cyto, and the relationship between Cyto and receptor architecture in cortical areas. And this is something I've been working on it for some time. And cytoarchitectonic borders correlate with receptor architectonic borders, so it gives us an additional modality with which we can analyze segregation in the brain, structural segregation in the brain. But it's not just structural segregation. Receptors also provide information concerning the functional organization of the brain and connectivity. And many people, when you talk about connectivity, they think about the axons and the fiber tracts and so on. But what they forget is that within the brain, signal is transferred by two mechanisms. Within a neuron, you've got action potentials, but whenever you reach the synapse, that the two neurons, they're actually not physically connected with each other. And then we have to bring in this chemical conduction, which is mediated by neurotransmitters and their receptors. And that's the way information can be passed on from one neuron to the next neuron, okay? And that's why looking at receptors also helps us find out something about connectivity. Now, if you look at this image here, it's different sections through, or coronal sections through a human brain in which we've labeled different receptor types. And you can see by the color coding where blue tones code for low receptor densities and red tones for high receptor densities, that receptors are heterogeneously distributed throughout the brain. You've got differences at the regional level, within the cortex, between cortex and subcortical structures, within cortex, within subcortical structures. We can see regional differences, we can see laminar differences in the cortex, and the research, which basically spans, it's basically all of my scientific life, has led in 2D, has led to identification of a couple of principles that we can associate with these differences in receptor densities. And that is that the heterogeneous distribution of receptors in the brain helps us identify cortical borders and also borders between nuclei and subcortical structures, that not all receptors reveal all possible borders, but some areas are grouped together by they build families of neurochemical structures, which is good, because this way these areas can build networks. We also know that the differences in the receptor densities, not when you just look at one receptor, right? You have to look at very many different receptors, so it is a multimodality. It's all receptors, but different receptor types, so you've got different modalities in there. They segregate functional systems. So if you look here, for example, S1 primary somatosensory cortex, it has a completely different receptor distribution pattern, receptor densities than M1 primary motor cortex. If we are within a given functional system, receptors highlight hierarchical processing levels. You've got gradations of receptor densities. I like gradations more than gradients, but this is what we've got. It's gradients within functional systems. We still don't know what happens at the whole brain level. Like in the sense of, do we have an overarching hierarchical system, that links the different sensory systems with each other? And then the nice thing, in order to enable translational neuroscience, is that these principles, they don't just apply to the human brain, which is the species that we used to define these principles, or which revealed them, but we have, it's conserved across species. And that's shown beautifully in this study, which we performed with Sean Frudist-Walsh, and in the macaque monkey brain, and one of the things that he did was to project the principal component scores of the first principal component onto the macaque monkey brain. And when you look at this distribution pattern here, when I looked at the distribution of the blues and the reds and the whites, it just reminded me so much of this drawing that Marcel Mézola made or published in his seminal work on the principles of behavior and cognitive, where you have this functional, sensory, motor, and associational cortices. So I'm very excited about the work that Thomas has been doing, and finally brought everything into 3D, which will enable us to do whole brain analysis. And with that, I pass the...
Great. Thank you.